So again, this is on behalf of the California Farm Demonstration Network. And real quick, in a nutshell, what that collaboration seeks to do is to showcase the implementation of conservation practices to provide a pathway for both new and experienced farmers. And it's also going to provide practical learning opportunities, at the same time fostering the formation of local connections through that network. Today is one of those learning opportunities. We're happy to bring you the first in the series of 4 about California soils in particular. I'm going to give you a little more information about that. The upcoming meetings in this series that we're calling the Nitty Gritty on California soils, it's going to be monthly. And so upcoming on April 21st, we're going to have Brent Holtz presenting on whole orchard recycling. We're going to have May 19th, Jessica Chiartis talking about soil sampling and how to get an effective sample that gives you meaningful results through time. And on June 16th, we're going to have Jennifer Moore talking about soil microbial dynamics. So if you're interested in registering for any of those upcoming sessions, some of you may have not had access for whatever reason yet, you can take a snapshot with this QR code right here, and it's going to send you to carcd.org's website, where we're hosting the registration for all 4 of these meetings. There's also a shortened website there on the bit.ly. If you copy that down, you can find our website, that web page directly really quickly. I'll give you all just a second to pull out those cell phones in case you want to snapshot that. All right, moving on then. This was advertised as making certified crop advisor continuing ed units available, and we are happy to do that. We're going to share a QR code with everybody at the end of the presentation, so you can immediately get those units. And if you don't use the QR code app for your CCA membership, then we're just going to take your number and name in the chat at the end of the meeting and make sure that you get those. Okay, so I am stopping sharing at this point. We've got a lot of people still pouring into this meeting. I think that the topic today has really struck a nerve with a lot of people. And so I'm really excited to see where the discussion goes today. So at this point, I think that I would like to go ahead and introduce our special presenter, Dr. Eric Brennan, who's with the U.S. Department of Agriculture's Agricultural Research Station, I think is what that S stands for, in the Salinas Valley. And at this point, Eric, I'd like to turn it over to you, tell a little bit more about what you do and give folks an idea of how the presentation is going to proceed. With that, you've got Zoom controls. Thanks, everybody.
Thank you, Sarah. So it's great to be able to have a chance to spend time with all of you today. So I work for the USDA Agriculture Research Service, which that's what the S stands for in their service. And we are one of the organizations of the USDA. There's also the Natural Resource Conservation Service and some other really good ones too. But the goal of the agency that I work for is to produce research information that helps farmers to become better at what they do, basically help them solve problems. And what I'm going to do in this presentation is it's going to be different than probably many other Zoom meetings that you've been to. So I expect that you are going to participate. And what I mean by that is if you don't engage with me, the presentation will be done in about 10 minutes. Okay, so I want you to ask questions. I want you to make comments. I may know the answers to some of those questions, or I may have an idea what the answer is, but I'm going to try to engage with you as much as possible so that this is a kind of a two-way flow of information, okay? So and the reason that I do this is that I think often in our presentations of research information, there's not enough time for dialogue or discussion. And so this is the format that I've been using for a lot of my talks lately, where what I do is I actually show a short video. So this video has been on YouTube for a while. You might have already seen it before, but I'll tell you what, I hope it makes you think about the cover crops and compost a little bit differently. And even if you have seen it before, maybe it'll bring up some questions that you hadn't thought of before. So that's how we're going to do it. It's about a 10-minute video. I will show you the video, and then it'll be played through my other screen here. And then right after that, I've got my my iPad here, and I've got a one of these Apple Pencils, and I've got screenshots of many different parts of that video, as well as some other potential graphics that I might use in our exchange. So I'm counting on you to come up with questions, comments, whatever. But I'm willing to be here for the entire time and try to engage with you, just like if we were in a classroom together and, you know, we could talk. Okay, so I'm looking forward to this. I've used this method with several other presentations, and from what I find, people, I think they like it. So let's give it a shot.
You know, before we go too far, Eric, I wonder if we want to take a poll real quick to get to know our audience and then start hosting some questions.
Hey, Eric, this is Karen. While folks are doing that, we've had several folks who are interested in looking at your data, kind of digesting it a bit in written form. Do you want to mention that they can find your published work on your website?
Yeah, in fact, let me start sharing my other screen, and I can lead you guys to where you can actually find that information really easily. So
I can tell you, if you just Google Eric Brennan ARS, you'll find it.
Yeah, that's right. Yeah, so basically, or if you just go Brennan Organic, you'll get to the ARS website, and at the bottom of the website, there is a, or somewhere in there it says publications. I put the PDFs for all of my publications on my website, so you can get the PDFs for those for free without any difficulty at all. Sarah, it looks like the poll is almost ended, or?
Looks like it. I'm going to rely on my colleague Kristen to help me close that out. My controls are giving me no access to it, so I'm just going to have to roll with it here.
Don't you worry, Sarah. I've got the poll.
All right. What does it say?
Well, we had about 85% of the participants on here fill it out, and top is in the other category, so that's quite interesting. Perhaps those of you selected other want to put in the chat how you would describe your affiliation. Second to that is, actually top is farmer, excuse me, 29% of y'all. Then other, then government, agricultural technical assistance provider. And then closer to the 10% category, we've got some researchers, nonprofit folks, and a couple ranchers and CCAs and PCAs.
All right.
Thank you all. It's so nice to have a nice crowd of folks here.
Wonderful. Thanks, Kristen. I'm glad to have you working with me today. So we have a little bit better idea of our audience now, Eric, and I wonder if there's anything you want to touch on first before we go to some questions, or?
No, I'm good to go right to the questions.
Great. So Kristen, maybe you can keep docking those in our doc here, and I'm going to go ahead and kind of go in order. And if I do also want to invite folks to, if you feel like you really need to do the raise hand function so you can verbalize your question, if it's a little bit more, harder to type in, we're open to that as well. All right. So I'm going to go back to early in the chat, and we had a question from a new landowner of 2 acres in the Mojave region with a goal to rejuvenate the land and eventually grow agave. I'm in need of guidance finding native cover crop to the Mojave. All the soil is super acidic and sandy. It says, haha. Darcy, I love the chat function, because Darcy responded by giving the Resource Conservation District's website. But Eric, do you have any thoughts on how people can get regionally appropriate cover crop?
I would do what the other person had suggested and go to see if there's an NRCS location close to you, or you know, cooperative extension through the university. I don't know anything about that area, so I'm not gonna pretend I do, but try to find, you know, local, state, or federal government agencies in that area, and they should be able to give you some help.
Yeah, for sure. I know that the partner institutions on our farm demonstration network include resource conservation districts, your county level NRCS folks, cooperative extension, the UC cooperative extension, and then there are other technical assistance providers who are able to perhaps help out with those specific questions. So I'm going to go back now to our chat questions, and Brianna asks, nitrogen leaching into groundwater being remediated by cover crop. That may be critical in nutrient management as part of Ag Order 4.0's coming regulations on the Central Coast. What do you suggest to do in case commercial fertilizer is adding too much phosphorus?
All right, so let me show you, let me go to the slide where I...
Oop. Eric, I accidentally just muted you. If you want to unmute yourself, I meant to mute my, thank you.
Oh, there we go. Okay. Yeah, so what I just pulled up here is this. Here's the visual that I used in that video to talk about the potential of these cover crops, with these guys right here, to basically cycle that nitrogen. So the nitrogen is coming out of our vegetables, and then it can come down into our cover crop and keep it from, you know, leaching down here into the groundwater. So the question was, what happened? It had something to do with fertilizers and too much phosphorus?
Yeah, it says, what do you suggest to do in case commercial fertilizer is adding too much phosphorus?
Oh, okay. Yeah, good question. So let me just write a few things here. So when we started our study, we were using a fertilizer 442. Okay, so that's the N, the P, and the K parts of the fertilizer. And this fertilizer is one of the worst ones in terms of an imbalance of phosphorus, because basically, you know, it's the ratio of nitrogen to phosphorus is quite high. The ratio, the amount of phosphorus in there relative to the nitrogen is quite high. So we use this for the first few years of our study, but then once I learned a little bit more about this issue, I then switched to another fertilizer, which is 811, which gave us a lot more nitrogen per unit of phosphorus, a lot less phosphorus per unit of nitrogen. So, you know, this fertilizer was one that really helped. You know, another thing that people could do would be to go to something like, you know, 1200, which is basically no phosphorus. I think that's feather meal. Now, one issue about phosphorus and organic fertilizers that's been quite interesting to me is that basically what I've noticed is that these materials here, hang on, let me change my, oops, back up there. These materials here, these like 442, I've noticed that the higher the phosphorus level in the material, the cheaper the cost of nitrogen. So basically, you know, if you're using something like 1200, you're going to pay more for your nitrogen. And this is one of the challenges. So, you know, if I was to draw a little graph here, if this is the cost of nitrogen, basically as phosphorus increases, so this is the phosphorus level here, the cost of the fertilizer basically goes down, of the nitrogen in that fertilizer. So this is one of the reasons why there's, you know, more interest often in using, you know, 442 than say 1200 or some other lower phosphorus material. So it's a cost issue here, and I understand the challenge of this, but ways to address it if economics are not an issue. I realize economics are an issue in agriculture, but would be to, switch to a different material. That's a very good question, though. Excellent question. And that's really what made me question this regulation, where growers can't use pure synthetic nitrogen. Because if you go and watch that video, what I'm talking about in the video is essentially, let me just zoom out here a little bit, but essentially all of the nitrogen in these fertilizers, or the majority of it, often is coming from conventional sources anyway. Yes, it's organically allowed, but it is coming from a conventional feedlot, or a feedlot that was, slaughtering animals that were fed conventional feed. So those nitrogen atoms in there did not come from biological nitrogen fixation anyway, and for the most part. They came from pure synthetic nitrogen that was used in a conventional field. It was fed to an animal, the animal then used it in their growth, and then those waste products got into the fertilizer. So very, very good question.
Wonderful. Thank you for that good question. I think Kathleen Beasley, looks like you have a quick follow-up perhaps to that.
I do. Thank you for taking it out of order. And so did you find when you switched to 1200 that you didn't need to use as much, because now you are putting more nitrogen in the ground? So would that offset the higher cost of 1200?
I think it was still more, we didn't switch good question, Kathleen. We didn't switch to 1200 because we actually couldn't afford it in our study. But we did switch to 811, and we can actually use, we're still applying the same amount of nitrogen, but we could do it, the concentration of nitrogen in that material is higher because it's 8% nitrogen rather than 4% nitrogen. And the bulk of the material is lower because basically it's more concentrated. But we did not switch to 1200. But actually switching to that 811 did reduce the phosphorus inputs, and that was one of our goals.
Neat. Thanks for that good question. Good use of the reaction icons too. Want to encourage everybody to do that. So Eric, unless you have a follow-up, I'll keep going down to the next question then. Okay.
Yeah, sounds great.
From Natalie, was there any breakdown of the microbial biomass in each treatment? Where were the differences in the species or genus of microbes present in each treatment? Really getting in the weeds here.
Okay, so let's go to the microbes here. So if I understand the question correctly, it has to do with whether or not we characterized the microbial community, the bacteria or the fungi, into different species. We did some general looks at this. And in fact, I'm going to show you kind of a cool feature of this program. So I don't have a picture of that, I don't think, here in this. I'm just quickly checking. I don't think I actually have a shot of that, but watch what I can do. So I'm going to quickly go down here to my computer, and I can actually quickly pull up the paper, and I have another nice graphic that shows, let me see if I can find where I'm at. Hold on a minute. Hang on. I just got to switch out of one program and into the other one. Let's see here. Hold on. Sorry, folks. This is why this process can be cool, because I can bring in other things, but it also may take just a half a second here for me to pull up the paper. I'm just doing that right now, and you can't see it yet, but you'll be able to see it in just a minute here when I take a picture of this. So
this is one of the perks of having Eric so tech savvy with us. He knows how to find what he's looking for real quick.
So just in case you're wondering, you can see my computer screen right there.
Yep.
So I'm taking a picture from this paper, and I'm going to use that photograph.
There you
And-
go. We see it nicely.
Okay, so let me now switch back to where I can actually see you guys. There we go. Okay. And let's zoom in on this image a little bit more. So this is from a paper that's I did with Veronica Acosta-Martinez, who's a soil microbiologist with the USDA in Texas. By the way, the link to this paper, as well as the one, this is the same paper where the microbial biomass data is from. There's a link in that video description to this paper. Okay, so whenever I refer to a paper in any of my videos, all you got to do is go down to the video description and you can get to that paper. So the question is, did we look at, say, differences in the microbes? We did, and we hardly found anything. So basically this figure kind of shows that. You can see here, here's our, let me kind of walk you through it a little bit. So we've got here's system one. Actually, let me just simplify a little bit here, because in the video, I actually only talked about these three systems right here, okay? So let's get rid of the rest of this here so we don't forget what we're looking at, all right? So these are the three systems that I talked about in that study. And this is the percentage of different groups, okay? So I realized I cut out the thing showing, the legend showing who is who, but basically what I look at when I see this is, you know, this pink group of which is one of the groups of soil microbes that were there, looks pretty similar across those three systems. So, you know, here's the system with no compost, and here's the two systems that got compost. This is the 1-3 that got compost and cover crop every year. You know, when I look at this data here, the size of this yellow set looks relatively similar, maybe a little bit higher over here, but you know, this pink grouping, pretty similar. Basically, our study did not show that we had big shifts in who was there in the soil in terms of the microbial community. What the study did show, though, is that the amount, the number, the biomass did change. And that's why, you know, let me get rid of that one there. You know, so if we go back over to this, hang on. Okay, so, you know, what I just said was who was there didn't change, even though the carbon inputs were very different. You know, system one here, it never got any compost and hardly was ever cover cropped. That didn't really change the composition of the microbial community. What it did change was how much microbial biomass, you know, the living bodies of those critters that were there. You know, it actually went down a little bit, whereas over here it went way up. So who was there didn't change much. And this is kind of consistent with, or is quite consistent with a lot of other studies that have shown it's pretty hard to see shifts, large shifts in the microbes that are present in terms of, you know, is it this species or that species or this group or that group? We don't know a lot about this yet, but what our data shows so far is who is there doesn't change that much. How many of them are there, or the biomass of that whole group, does change with our management. Mm-hmm. This is one of the reasons why I'm often very, very skeptical whenever I hear about these inoculants that can be added to it. Like, you add this inoculant and boy, it's going to just change your soil. You know, I would call these bugs in a jug. I'm often quite skeptical that there's going to be much benefit to that. You know, the soil is a huge reservoir. Adding a little bit of inoculum often has essentially hardly any effect, other than maybe a few things like maybe mycorrhiza or rhizobia if you're inoculating legume seeds. Anyway, that's probably enough on that question.
That was great. A little bit of a follow-up from Nellie is, what did you use to quantify the microbial biomass?
So we used a fumigation method, and this is a great question, because guess what? It's pretty hard to find that for farmers to get access to that. So I actually was wondering the other day, what could farmers do that could actually give them a good measure of microbial biomass? Because the method that we used was kind of a nasty one. It's very widely used by researchers, but here's a system. I came across this just the other day, and I have not seen this used in vegetables, but it was an intriguing idea that I thought would be worth looking at. So this is a study by Jennifer Reeve and her colleagues at Utah State. And what they did was they actually used basically a kitchen sieve. They took that kitchen sieve, so, you know, one of those colanders like you put your pasta through. They dug up soil from an orchard, and they stuck that soil in this sieve, and then they do what they call a modified slake test. You've probably seen these on YouTube. You know, there's many different people who've talked about them. It's where you put soil into some kind of a holding container, then you stick that soil, you know, like a screen into the water, and you watch the soil particles either fall apart or stay adhered to each other. It's quite visual. Anyway, this modified slake test, which you can learn about if you go to just type in the name here of this paper, simple soil tests for this orchard system. You can get access to that. But here's the beauty of this. It's simple. And for their orchard system, it was highly correlated with microbial biomass carbon. And this figure here shows you that. So, you know, let's just look at this real quick so we make sure we're on the same page. This is microbial biomass. That's what I measured in my study. They also measured it in their study. But what they did was they also used this fancy kitchen sieve modified slake test. And check it out, folks. The correlation, you know, there's a nice trend there where basically as the microbial biomass goes up, so does this percent stable aggregates, which is what we've got here on the y-axis. So, you know, in fact, you know, I've kind of covered it over here, but a correlation of an R of 0.83 is excellent. That's very well correlated. Now, what I don't know is, would that be a good measure of microbial biomass in a vegetable system? I don't know that answer. I wish I had known about this test earlier, because I would have liked to have tried it out in our 3 different systems where we had large differences in microbial biomass. So that would be a test that I would consider using. Now, here's another cool thing about this test that they did. They also measured, you know, what are the growers like in terms of a soil biology test? They like this sieve method. And, you know, I don't want people to waste a lot of money sending their soil off to labs that say it'll have, you know, this amount of ratio of fungus to bacteria and this or that. I would say be very cautious with spending money on some of those tests until we've got better data on that, because I think often simple tests are the best. And I'll tell you what, the best biological test that I've seen, look at your crops. You know, I mean, look at the crops in our study, the lettuce yields. That is a biological test of how that soil is functioning. You know, if I go back to this slide here of, let me see, where was it? This one of, hang on a minute, I just gotta find it here. Results, there we go, this one here. I mean, check it out, folks. This right here, this high number in terms of my lettuce yields for a system that got cover cropped every single winter, that is a measure of soil biology right there, because yeah, I'm not saying it's microbial biomass, but it's a whole combination of indicators of whether or not that soil is functioning well or not. And to me, that's actually one of, people think about these other tests, but what about just using your crop as a measure of soil health? Keep it simple. Don't waste money on things that we don't know as much about. Let scientists do that part and then slowly start to learn more about that. I will, there's a paper which I don't have at my fingertips, but if anybody sends well, let me look it up and I'll read you the name of it at least. I'm just trying to think here for a minute where I can, I can't think of the name of it right now, but it's a study that was done recently where they looked at several different soil biology tests. And they basically, this was, I'm trying to think of the name of the scientist. They suggested that farmers not generally measure soil health using these lab analyses. So that was sort of a general comment, is use other more standard soil tests. Don't waste a lot of money on soil biology or soil health tests at this point.
Interesting, Eric. That's a good segue that, into this next interesting question from Evan. And he says, placing a sock in soil and monitoring deterioration rate is also a good way to measure microbial activity, right? Thoughts on that?
It is, in fact, so I'm on Twitter. If you go search for me on Twitter, I posted something after I found this thing about the that modified slate test. I also learned about the underpants test, and I'm not sure that one has been well researched, but it's an interesting idea. I actually posted a thing on Twitter a few weeks ago about that. Basically, the idea is they take a pair of white men's underwear, and they put those white undies in the soil, and they watch and see, how degraded do the underpants get over time. Now, I don't know if there's a lot of complicated things there. It's not quite as straightforward as if the underpants fall apart in a week or two, you've got a healthier soil, because it does depend on, soil moisture, whether or not what type of crop you've got in there, a whole bunch of other things. But in my tweet about the underpants thing, which caught my attention, I added a link to a video by another person in Canada who talks about some of that complexity. So now there's no reason to think that a cotton sock would be any different than a cotton pair of underpants. But that's a neat idea. There was a thing at UC Davis at this soil, one of the sites that I think I included there from UC Davis in the tweet. It talked about if you soak the underpants in glucose, it actually speeds up the process. Now, I wish I'd like to try this in a vegetable situation, because maybe that's all we need. I don't know.
Thanks for that, Eric. And it is nice to have those kind of simplified tests and rules of thumb to work with. I'll go to the next question from Robert. He asks, can you tell us a little bit more about the compost applied? Where did you get it? Was it tested? And if so, what were its key properties? And remind us the application rate, and what you would say are the key impacts, carbon, microbial boost, phosphorus, nitrogen, etc.
Sounds good. Okay, good question, Robert. All right, so here's that compost, and here's the application rate, 7 metric tons per hectare. Now, one thing I didn't mention, let me just quickly jump over to another slide real quick, just to make sure we're on the same page. So, in all my data, because in science, we generally we use the metric system, we don't use pounds and acres. When all of my data is published in kilograms per hectare usually. but if you want to convert kilograms per hectare to pounds per acre, just multiply by 0.89. Okay, so keep that in mind. So they're relatively similar. Okay, so and by the way, you know, 1,000 kilograms per hectare is 1 metric ton. Okay, so let's go back over to that other slide where we've got the compost. This one here. So basically, this compost here is 7,600 kilograms per hectare, and if you multiply that by you know, 0.89, then you'll get how many pounds per acre that application rate is, and you can convert that to regular tons if you want. So that's the application rate. Where did the compost come from? Our compost came from Z-Best in Gilroy. The compost actually, here's a, boy, I was kind of thinking ahead, wasn't I? I had a few different slides ready for you guys just in case you ask about this. So what is the source of that compost? Well, let's check it out here. This compost is basically from yard waste. Okay, so, you know, this is actually yard waste being picked up in my neighborhood. So that yard waste goes to Z-Best. They grind it all up. It's tree limbs, it's leaves, it's prunings, etc. It's ground up. It goes into these big windrows, like right here. So it's aerobically composted, turned over several times, you know, until it gets to a certain point of maturity. And then the material is actually screened right here. You can see it being screened through one of these things. And then it's sent over to the farms or it's transported to the farms where it's used. So let's go back over to this other one here. So that's the source of it, okay? So Z-Best was our source. A little bit about the characteristics of the compost. Probably the most important characteristic, I would say, is the C/N ratio. So the C/N ratio of our compost is about 20, you know, it's somewhere from about 18 to 22. I think we estimate on average it's about 22. So that's the carbon to nitrogen ratio. That's how much carbon atoms there are in there relative to the nitrogen ones. Anything about the microbes? I don't think it matters. I don't think microbes in compost matter at all, basically. Now, some people might say, hey, I thought, you know, you're adding microbes to the soil when you add compost. Yeah, you are adding some, but think about all the microbes that are already in the soil that are way over diluting any microbes that are added through compost. So you get some microbes through the compost, but do they have any effect? I would say probably not, because if you remember this system here, you know, this one here never got any compost. And we didn't see a big shift in our microbial community at all. We saw a shift in how much microbial biomass there was in the soil, you know, the living part of the soil organic matter. But we didn't see a shift in, you know, say more bacteria or less fungi or whatever. That didn't change. So does that kind of answer the question?
I think it does. If we need to follow up on that, go ahead and raise a hand right now. I'll look for it over in my screen. Not seeing a raised hand. You can always follow up in the chat. Kristen's keeping up with y'all. So thanks for that, Eric. And there's some really good discussion about the underpants test still in the chat. Some information sharing. So that was interesting to folks. I'm going to say we've got a question here from Eric, and he says, I have a small scale market garden. We use compost and have a crop in the ground year round. I'm curious what you think about this. We don't cover crop, but there's always a crop in ground. However, that crop doesn't get incorporated, but we do use a fair amount of compost. So I think it's kind of general thoughts on that. If you have crop in the ground year round anyway, what might be the relative benefits or lack of by not having necessarily a cover crop?
Yeah, so, I think what you're saying is, if you had, say you had lettuce or let me back up here. Say you have lettuce for a while, and then you go to broccoli. Sorry about my writing. Then you go to kale. Something like this. So you've got vegetables year round, and you're not growing a cover crop there. I can see how this is often a common practice, especially where farmers are more limited in their land area. And whether or not it's a good practice from an environmental standpoint may differ from whether or not it's a good practice from a groundwater standpoint. So, if you're adding a lot of fertilizers or compost in these systems, one could easily overload the system with phosphorus. I am concerned sometimes. I've heard of some systems, where they'll come in and they'll add several inches of yard of green waste and get a really thick layer of that going. That's adding a lot of phosphorus to the system. And if you have any runoff from that system, you can have a lot of losses. By not having a cover crop in there, if you get a lot of rain, you could have leaching, but maybe some of your vegetables that you're growing during the wintertime have a good deep root system. So maybe that's not as much of a problem. It didn't sound from the question like maybe there was as much tillage. And so, when we reduce tillage, we often will reduce the rapid release of nutrients, from these systems. So maybe that's not quite as bad as what we're seeing in our systems where, if you remember, I'm trying to remember if I think I got a picture here somewhere of it. Our systems are highly tilled, so tillage exacerbates nutrient release and can exacerbate nutrient loss in some of these systems. So, whether or not you're having a loss of nutrients in those systems or not, I don't know. But I do like adding diversity from the cover crop, in our systems. The cover crop is a grass, very different from our vegetables. The cover crop is also able, let me just back up and go to a different picture real quick here. One of the beauties of a cover crop, if we were to take the soil, let me just kind of draw a little bit of some soil here. So let's say this is our soil up on the top here, kind of the rooting depth, say it's to 1 to 2 feet. And then you've got a bunch of other soil down here. Often vegetables are relatively shallow rooted. And so you may have a whole bunch of nitrogen that's coming from this, in this top zone that's coming down here that your vegetable root systems, can't access. And if you put a cover crop in there, let's draw it like a yellow one, like, let's say there was a cover crop in there like rye. Rye's root system is extremely deep. And so it can often get way down there and hopefully scavenge and bring up some of those nutrients. Now, there are some vegetables, my colleague Richard Smith has done here, he's with Cooperative Extension here. He's done some interesting work with some vegetables like some of the brassicas, which are actually quite deep rooted. So bringing up nutrients from deeper layers is one of the benefits of cover cropping. The other thing you might just consider too is, when you're having all that vegetable leave your land all the time, your actual input of carbon that you produced on your acreage is relatively low. So what you're really doing in the system that you're bringing in lots and lots of compost is you're relying on somebody else to grow your carbon. And this is an issue from a sustainability standpoint. I think sometimes we need to think about carefully. And I'm going to go to a slide here, which hopefully will make you smile a little bit. I don't know if you like, what do you call this? Connect the dots. So one thing that I've sort of concluded from our study, if we connect the dots, is that with compost, let's see, let's pick a red color here. Now I'm just connecting these dots here. I have a big question mark about compost. I can see some benefits from it, but I can also see some challenges. I have no questions at all about cover crops. All of our data consistently shows that cover crops are beneficial in these systems. Our data does show that compost can be also, but I'm concerned that we can over-apply nutrients easily with compost. Using a lot of compost gets us away from producing any of the carbon that our systems need on our farm. So, you know, a place like Salinas Valley, we've got lots of organic farms here, but unfortunately, a lot of those farms are not hardly producing any of their own carbon that is other than vegetables. Okay, so and that's because cover crops are infrequently used in these systems. And yes, compost can definitely improve the tilth of the soil and add nutrients and things like this. But without a cover crop there, you're basically wasting all that sunlight. You know, that sunlight is critical to try to produce carbon or energy on farm. Now this is kind of a long-winded answer, but I'm going to tell you about a really, really inspiring scientist who writes a lot about this. His name is Janzen. If you want to read poetic science writing, you've got to read Janzen. He's a soil scientist in Canada. He writes the most beautifully written papers I have ever written. Here's one of my favorite ones that he, the soil carbon dilemma. Should we hoard it or should we use it? It's eloquent. I mean, it's so good. This is one of my favorite ones. It's from 2006 or so, I think, or 5. Another one of his that I spent a lot of time reading this past week is let me just zoom in a little bit more. Beyond soil carbon sequestration, soil as a conduit for for solar energy. So, he talks about how we need to come up with ways that we can maximize the energy going into our systems with carbon. And so, when I think about cover crops, and we go back to this shot way up here at the beginning, this system here, where farms are relying on a lot of compost from green waste. I know that's common, but the cover crop, when it's grown as a replacement for compost, is providing so many services. We call those ecosystem services. It's reducing nutrient leaching. It's adding diversity to the rotation. It's reducing runoff from the field. It's providing habitat for beneficial insects. It's adding a fresh source of organic matter that is quickly cycling in the system. Those are all really, really, really important things that compost just doesn't do as well as cover crop. If you go and look at the amount of inputs that we had in our study here, check out in this figure here, all this input, most of this right in here is just compost. Yeah, we've got a little bit of cover crop there in system 2 here, and a little bit there. But remember, that system didn't perform very well in terms of yields. We could have increased the yields of our lettuce quite a bit in that system just by pumping up the fertilizer level. But that has a cost also, right? Anyway, so that's kind of a long-winded answer to the good question you had there.
Yeah. No, that's some important considerations, and really making the case for how it sounds like you can't really go wrong with the cover crop route.
There are some challenges which we can talk about if you want to, but let's go see what the next question is.
Sounds good. Sounds good. Yeah, keep those questions coming, folks. I've got one here from Mary Chambers. She says, you mentioned adding pure nitrogen fertilizer in organic systems. I wonder how you expect that would affect nitrogen runoff and oxidation into nitrous oxide, a powerful greenhouse gas. Also, how do you think the energy input required to produce synthetic nitrogen compares to the energy input required to produce an amount of compost that would provide equivalent support for plant productivity?
Yeah, there's a lot there. Let's go to let's go right here. Okay, so start with it. Read the first part again to me, will you?
You bet. You mentioned adding pure nitrogen fertilizer in organic systems. I wonder how you expect that would affect nitrogen runoff and oxidation into nitrous oxide, a powerful greenhouse gas, since that nitrogen is not bound up in anything.
Yeah, it's a good question. So when you add nitrogen, whether or not it's in organic form, like this right here, this pelleted material, or in a pure synthetic form like that, the fate of that nitrogen depends on how much you apply, when you apply it, how you manage your water in those systems. And if you're managing it carefully, you can have, I think there's a lot of evidence to show that you could have good nitrogen management from synthetic material or from organic, or you could have bad management from either of them. So it is true that the nitrogen in the organic material is in a different form. It's got to go through mineralization quite often to release that nitrogen into a form that's nitrate or whatever the plant is taking up. And so the chance for leaching there probably at the beginning is lower. But we can have lots of leaching from tied up nitrogen in organic forms also. And probably some of the best evidence of that is, all the nitrogen that's in our vegetables at the end of the season, say broccoli, that's tied up in biomass. And yet we turn that under, and we can have a huge loss of nitrogen as that material decomposes. And that's why we have to have, if we want to manage that nitrogen carefully, we have to have a cover crop there to grab that nitrogen before it gets below the root zone. So I can see how pure synthetic nitrogen could be used very sustainably, or you could do the same in terms of management with an organic material. So in terms of green, the nitrous oxide, that often really depends on say the how aerobic or anaerobic the soil conditions are. I've not seen evidence that would suggest that if you're applying the same amount of nitrogen, it would be any worse or better with organic or conventional fertilizers. I can see how it could be bad with either one. If you haven't managed it carefully. It's a little more complicated than that, but the management is, I think, more important than the material, quite honestly. you know, there is actually some evidence in the literature that shows that cover crops, legume cover crops, sometimes can increase some greenhouse gas emissions. There's a paper that's, it deals with this. I think it's by Casper. So, you know, non-legume cover crops tend to be a little bit better in that regard, but it's complicated. The other part of the question was the second part.
You know what? I've got Mary Chambers here with her hand raised, so I'm going to let
yeah.
her re-articulate her follow-up question. Does that sound good, Mary? There you
Hey,
go.
Mary.
Oops, I clicked you to unmute right when you were unmuting yourself. There you go. Go ahead and unmute it.
Thanks. I was just wondering, Eric, you mentioned that you haven't seen any evidence that nitrous oxide release is higher from synthetic nitrogen application than from nitrogen application in compost or organic material. Has there been any studies on that? Because I think that would be a really interesting direction for research. I mean, absence of evidence isn't evidence of absence, or whatever they say. And then I guess my second part of my question was just, because the energy input for creating synthetic nitrogen fertilizer is so intense, I wonder, but compost production also needs energy input with the mechanization of compost production. And I was wondering, how do you think the energy input required would compare between producing a certain amount of nitrogen fertilizer that would lead to a boost in plant productivity versus a quantity of compost that would lead to a similar boost in plant productivity?
Yeah, these are great questions, Mary. So, you know, regarding the nitrous oxide part with, I want to make sure I didn't overstate it. I think some studies have been done on that, but I can't pull them off the top of my head. I have to go look at my database to find them. But I don't think I would be as worried about the material as I would be about the way it's managed. Regarding the energy costs, this is a huge issue. In fact, let me tell you something first just about the repackaged synthetic nitrogen part. Keep in mind that all that nitrogen that is in this repackaged form was synthetically fixed, or the majority of it, because it came from conventional feedlots. So the cost of that is already, you know, we've, organic farms in this area are relying on that synthetic nitrogen, which is energy intensive to produce. They're relying on it though, after it's passed through animals and then it's allowable. So just keep that in mind. But this issue of, say, compost, you know, you raise a really important one here. So this process to get this, you know, if we go back here, the cost of getting this green waste to the compost facility, that's all fossil fuel driven. The cost to get it over to windrows, that's all fossil fuel driven. And then to bring it over here, it's again fossil fuel driven. So there's a lot of costs with that. There's also an interesting thing that happens with compost. During the composting process, about 50% of the carbon in the material actually goes off as CO2. So what I would really like to see is a really, really good study in California that looked at what's called the life cycle analysis of compost. And now, you know, don't get me wrong, I'm not for reusing, I'm all for using green waste. But as a source of nitrogen, it's, you know, there's some issues with it. You know, so when I say life cycle analysis, I'd like to see, you know, this cost right here. What's that? Also this part, and add it all together and just see, you know, is it any better or any worse? Recycling is great, but you know, the other thing we often don't think of is when the compost leaves a lawn or somebody's yard, you're basically removing nutrients. Those have to be replaced somehow, or over time that system could actually decline. And so to do that, people are going out and, well, I'm go to Home Depot and I'll buy some more fertilizer so I can grow more grass. that fertilizer, for most part, is coming from synthetic nitrogen. So your question is great. I'd love to see a full life cycle analysis of compost, the costs, the benefits.
And Eric, on that note, I just noticed that Stephen in the chat referred to Professor Alyssa Kendall in the Ag Sustainability Institute at UC Davis, does a life cycle analysis, has some data on the compost. So an LCA on compost there. So thanks for that.
I'm gonna look that up. That sounds good.
Yeah, love the activity in the chat room. Mary, great questions. Before I answer the raised hand here, I just want to look at the time. It's 12:21. We're planning on wrapping at 12:30, at which time I'm going to share that QR code for the CCAs in the audience to get their continuing ed units. It's been a great discussion so far, and we're just going to keep plowing ahead. We'll try to get through some more questions before the end. With that, I'm going to ask Zahangir Kabir to articulate your question. I hope I did not pronounce that right, but did my best. Go ahead.
Yes, hi, Eric. How are you doing?
Great, Kabir. Great to hear you.
Yeah, it's a great presentation. Yeah, I have a question about you said the nitrogen fertilizer. You said this can be used in organic system, but if you're looking at the history of nitrogen fertilizer, in 1960. Efficiency of nitrogen fertilizer was 75%. Now it is actually below 25%. So the efficiency of nitrogen fertilizer actually is really low. Other thing is that, especially in vegetable system, let's say lettuce has a very small rooting systems, and efficiency is going to be even much lower if you are applying synthetic nitrogen fertilizer in the system. Thank you.
So when you say efficiency, what can you expand on that a little bit, Kabir?
I saw an article by Tillman in Nature. He actually worked on that nitrogen efficiency of cereal productions. And they mentioned it in 1960, efficiency of that nitrogen for cereal production was 75%. But now, just for the cereal production is actually 25%. So it means 75%. Once you are applying 100 pounds, 75 pounds actually going to the maybe going to the water body or to the atmosphere, and 25% can be used by the plant. And so it's gradually actually reducing the efficiency of nitrogen, especially chemical fertilizer.
So you're saying the nitrogen use efficiency is going down over time?
Yeah, that's the history, 1960 to 2000 actually.
I'd like to see that, because I'd like to see that paper. Maybe you can put it in the chat there. That sounds intriguing. One issue to think about and this is sort of related to Kabir's comment, is just in general, is what are the alternatives to using this these feed waste or feedlot fertilizers, the pelleted organic fertilizers? What are the alternatives? Well, one alternative is to basically grow legumes, synthetic or biological nitrogen fixation. And that's that was the method that was used. And I talk about this a little bit in the video, but that was the method that was used up to, the beginning of the, say, early 1900s. But most of us wouldn't be around if it wasn't for synthetic nitrogen. And there's just not enough land area in the world, from all the analyses that I've read, to produce all the nitrogen that we need from biological fixation. We just have too many people. Our planet is basically, most of us are here because of synthetic nitrogen. I mean, we're here because of our parents, but we wouldn't have had enough to eat if it hadn't been for that. And so, if we had to rely totally on biological fixation for our nitrogen needs, we would need to take a lot more land area to fix all the nitrogen that would be needed biologically. And so that's why what I would love to see is more work on renewable energy being used for synthetic nitrogen production. But there's also another issue that's a challenge with using legumes in, say, vegetable systems, and that is the synchronicity of release. So, if you take a timeline here, you incorporate a legume cover crop, it releases its nitrogen pretty quickly. Say this is the nitrogen release, and here's your vegetable nitrogen demand. Those two are not overlapping, they're not in sync. And so this is a big challenge with trying to get them to kind of dance together so that they're not stepping on each other's feet. And this is one of the biggest challenges with legume use in these systems is trying to time that release. Is not very easy. And that's why we use fertilizers often, even whether or not they're organic or conventional. But in our system, if I can kind of back up here, this picture here, the amount of biological fixation that we found in our study, that's not enough to meet our vegetable nitrogen needs. This pink area, that was a legume grown every single year, every single winter. And it's not producing enough nitrogen for our vegetables in terms of the quantity, but also the release is not well timed with the release in our systems. And this is one of the biggest challenges with these systems is trying to figure out how to get them to synchronize right, as well as produce enough. This process, by the way, this nitrogen fixation process is extremely energy intensive for the plant. There's a really good book written about this. I think it's called Enriching the Earth. The author is Smill. So Enriching the Earth. The Earth. And it talks about the energy intensity of biological fixation is extremely high. The plants don't just do it for free. They've got to get a whole bunch of carbohydrate in return. So anyway, I'm going to check out that paper, Kabir. Thanks for posting that.
Yeah, and other things, root in architecture is another area probably, breeders should work on, especially lettuce, broccoli, all those vegetables. Root system is so small. So the efficiencies, the nutrient, acquire is less, in that kind of vegetables.
Yeah, Louise Jackson, who's a emeritus professor at Davis, did some really interesting work several years back in Salinas Valley looking at the root architecture of lettuce. And it's an area where I think more can be done to try to improve the scavenging ability of some of our vegetables. It's an interesting area. I haven't done any work with roots. Roots are very hard to work with because they're underground. But what we can do is we can estimate, root sort of biomass. In fact, if you want to read one of our papers, I'll just quickly show you just to kind of maybe entice you to maybe go and check this out. So this is from a paper. It's this paper here that we published just last year in the journal PLOS One. And in the paper, we actually show this really nice figure that I really had fun putting together. This shows the carbon inputs below the ground that we estimate from the cover crops as well as the vegetables, as well as the carbon inputs from the shoot of the cover crop, as well as the residue from the vegetable. So I don't want to get into this too much, but if you want to go check out that paper, there's a pretty figure that'll hopefully draw you in.
Wonderful. Eric, we've come near the end of our time. Kabir, if you had any thoughts to wrap up, I didn't want to cut you off before I move the presentation on.
That's okay. Thank you so much. You know, Eric, always great presentations and wonderful videos.
Yes.
Oh, thank you.
Eric,
Sarah, just a quick question. Did we get to most of the questions, or were there still a whole bunch that we never dealt with?
there were still a bunch of questions. We got to about half. And what I would like to, I wonder, Eric, if you could work to stop sharing your screen. I'm going to transition into that QR code pretty soon. But what I would like people to know about Eric's videos is that he is actively engaged in the comments on each of those videos and provides really great resources and checks that pretty frequently. So I'm going to encourage everybody on this line to check out Eric's YouTube page. And if you have more good questions, you can engage under those YouTube videos. Eric, you want to remind people what your YouTube page is called?
Yeah, so if you just go to Google and type in Brennan, Brennan, my last name, organic, the YouTube channel should come up and you can find the video that we saw today. And yeah, I do want to emphasize what Sarah just mentioned. Part of the reason I make videos is because I want to interact with people about the research. And one of the best ways to do that, other than as something like this, is to put questions into the comments about the video. And then I will, first, you put the question in there, the question won't show right up. I'll see it first so that then I can check it and say, all right, this is a question there's, no inappropriate speech or something in there. I moderate all my questions. But if it's anything is a good question, I allow it on there and then I try to address it. Sometimes it may take a few times back and forth before maybe we get it right, but it's a fun way to engage with people. And I want to thank you all for engaging. This was fun. I hope you think this was a worthwhile use of your time. Maybe better than if I had just prepared a whole bunch of stuff in advance and didn't give you many choices, but I enjoyed it, so thank you.
You're getting a lot of thanks coming in on the chat. I want to thank everybody for joining us today. This was really interesting. This chat series of questions is very valuable to us, and I just want to let everyone know that as the current coordinator of the California Farm Demonstration Network, I'm going to hold on to that list of questions, and I'm going to route them to the right people who need to see those questions. If you're asking for research, and if I can steer those good questions toward our partners at the UC Ag and Natural Resources or UC Davis or NRCS, I'm going to do that. And I want to remind everybody that we do have 3 more upcoming meetings where we're really digging in on the nitty-gritty of California's soils. The Farm Demonstration Network really seeks to give you California-specific information, keeping it regional, keeping it crop-specific when we can, so we can really get you the information you're looking for. So with that, I'm going to go ahead and share my screen so those of you who are CCAs in the audience can take a snapshot of the QR code. Let me know if I've never actually done this before, so if it's too small, let me know. Maybe I can make it bigger or something, but I think you all can see this right now. And if you are not a CCA that uses the QR code, please go ahead and enter your CCA ID number in the chat with your name, and I'll send those over to the certified crop advisors and make sure you get your units. So I'll just kind of hang out here for a minute and let you all enter your codes. Bring my chat back up. Get little thanks for that feedback, Ian. Appreciate it. It looks like the QR code thing is working. And yeah, I'm going to be posting this video, this recording, on the carcd.org website. I'm going to be updating the project page. You're going to find the California Farm Demonstration Network under the current projects part of our webpage. I'll be updating that soon. I'll put a slightly modified version of this talk in there so that I can encourage you all to go straight to Eric's YouTube video first and engage in his comments and questions on his YouTube video before playing back the rest of this recorded session. Kristen, can you do me the favor of downloading, saving the chat room function and letting me know you've got your hands on that file? I'm going to go ahead and I think I can stop share now. I like seeing everybody docked on my screen to see who's left.
Sarah, I've done a cut and paste copy of the chat as well, and I'll share that right away with you. Eric, there's a lot of really nice comments for liking your approach, and also a lot of questions. I threw my email in the chat and told people to go ahead and reach out to me, because a lot of questions seem to be looking for some resources as one of them that you took. So Sarah and Kristen and myself and you, as you know, as appropriate, can try to connect people.
Wonderful.
Sounds great.
Yeah, I want to encourage people to reach out to me. I dropped my email address in the chat a while back, sarah-letton@carcd.org. You can find me on that website too, and link to me. I want to take this opportunity to give some due credit to Karen Lowell there, my buddy at the NRCS, who really helped me sculpt this meeting series to dig in on the California soils, and really was able to articulate what it is that a lot of you are wanting to know about, hence the excellent turnout on this presentation today. So thank you so much, Karen. Thank you so much, Kristen, helping me manage those great questions in the chat, and for always being just a great support in this project. And finally, Dr. Eric Brennan, thank you for a wonderful presentation, highly engaging. We had great attendance all the way through to the end here, which is always great feedback too. Mary's clapping, thank you so much. And Karen, Kristen, thanks for capturing that chat, and we can reach out to folks. And lastly, I'm just going to go ahead and plug in my email address in case anybody wants to be added to the California Farm Demonstration Network listserv that I'm putting together. I'm grabbing those email addresses so that when good opportunities come out from our collaborative organizations, that you'll be the first to know about it.